Reemergence of monkeypox, prevalence, diagnostic, and countermeasures. Introduction. As the world is still recovering from the widespread COVID 19 pandemic and coming out of trauma, another outbreak known as monkeypox virus strikes. Human monkeypox is a rare viral zoonosis endemic caused by the monkeypox virus, a member of the genus orthopox virus. Human monkeypox is clinically almost identical to ordinary smallpox, and therefore, since the global eradication of smallpox in 1977, much attention has been paid to monkeypox as a smallpox like disease and possible agent of bioterrorism. The clinical picture of monkeypox closely resembles to the one of smallpox, but the major difference distinguishing monkeypox from smallpox is the lymph node enlargement that occurs early, often at the onset of fever. Monkeypox can infect a taxonomically wide range of mammalian species, but the natural host is unknown. Monkeypox is an emerging zoonotic disease recognized as the most important orthopox virus infection in humans in the smallpox post eradication era. It manifests with the same symptoms as smallpox, including flu like symptoms, fever, malaise, headache, back pain, and characteristic rash.
History. The first case of monkeypox virus in humans was recorded in 1970 August in the rural area of Democratic Republic of Congo. It was a 9-month-old child who was admitted as a suspect of smallpox infection and his sample was sent to WHO Smallpox Reference Center Moscow, where it came out to be a monkeypox virus as a result of virus isolation. The first monkeypox virus case in Nigeria was reported in 1971, and between 1971 and 1978, 10 monkeypox virus cases in Nigeria were reported. A total of several thousand human monkeypox cases have been confirmed since then, with 11 of them occurring in countries in Africa. Ever since, there have been emerging cases of monkeypox virus every now and then, but 1981 created chaos as it registered the greatest number of cases in West Africa. Another outbreak was seen in 1996 and 1997 in Democratic Republic of Congo, having an infection rate of 22 cases per thousand population. In 2003, the virus was unstoppable and migrated outside of Africa. The first report was filed in the United States because of the shipment of rodents imported from Ghana to Texas.
Pathogenesis. Monkeypox virus is a double-stranded DNA virus and the most dangerous member of the orthopox virus genus, the Poxviridae family, and the rest of the other member of this genus are vaccinia virus, variola virus, and cowpox. They all are pathogenic to human. Monkeypox virus infection starts with dermis or respiratory epithelium following transmission from an infected animal or person. Dispersion of the virus occurs through the lymphatics to the blood with primary viremia and systemic infection. Secondary viremia occur and result in infection of the epithelium with resultant skin and mucosal lesions. Replication of the virus in mucosal surface can cause in its transmission via oropharyngeal secretion to close contacts.
Clinical manifestation. An average of 6 to 16 days is required for monkeypox to develop during its incubation period, which ranges from 4 to 21 days. Then comes the prodromal illness, which lasts for 1 to 5 days. Asthenia, acute myalgia, back pain, and lymph node enlargement are some of the typical symptoms of the prodromal illness. Skin rashes usually begin on the face and later spread to other parts of the body, including the palms and soles. The lesions spread centrifugally from the face and extremities to the trunk and genitalia.
Diagnosis. The diagnostic test for monkeypox virus is similar to that of the orthopox virus test, such as viral culture, isolation, electron microscopy, immunohistochemistry, PCR, including the real time PCR, etc., are proved to be very effective when studied along with proper clinical and epidemiological report. Viral DNA is extracted and two real time PCRs are done to detect the presence of monkeypox virus DNA.
Treatment and prevention. The final step in the global eradication of smallpox was the combination of vaccines and a vigorous vigorous surveillance campaign. However, vaccination with the vaccinia virus is quite effective at preventing monkeypox virus infection. Pre exposure vaccination is advised by the Center of Disease Control and Prevention for anyone who has direct contact with suspected monkeypox virus infected animals. Laboratory workers who handle monkeypox virus possible specimens, investigators of animal or human monkeypox cases, caregivers of monkeypox patients, and others. An oral equivalent of the injectable medication Kidofovir, Brincidofovir, and many have better safety profile than Cidofovir, including less renal damage. FDA approved hyperimmune is used to treat some vaccine related side effects. Since June 2021, Brincidofovir has been authorized in the US for the treatment of smallpox.
Conclusion. In conclusion, monkeypox virus infection is relatively rare and usually self limiting. With increased globalization and cross border movement of animals, monkeypox virus can spread to different parts of the globe. The symptoms of the disease in human can be very similar to those of smallpox, chickenpox, or other causes of vesiculopustular rash. Therefore, accurate and rapid laboratory diagnostics are paramount in controlling an outbreak. In particular, we thank you.